Welcome to the Namasivayam Group.
Welcome to Genoverse, where we decode and recode the genetic switches. We are trying to make our journey a small step towards the personalized medicine, where the current evidence-based medicine has already minimized the failure and adverse effect. We try to integrate the genetic and the programmable approach together and try to devise a strategy to target the right place, not at the cell level, not at the protein level, but at the nucleic acid level. Also, we try to know what is the right time to target. So to do so, we particularly focus on eye research. This particular photo taken by my PD, Joshi, shows the ICMS in day and night. So day and night can have effect on our moods, on our organs, and at the molecular level, the gene expression. We have used the machine learning approach to make a first comprehensive circadian atlas of human retina that can guide us to target the right gene at the right time, where the expression is higher here and not here. Thanks to Yusei Sensei, now you know the meaning of my name, Namasivayam, where each syllable represents the 5 elements that makes up the universe. Like that, the Genoverse is also represented by the 5 elements. I want to categorize that. So where the Na, here, where we work on silicon nanoparticles recently, and Ma, water, where we try to make use of the lateral flow for the diagnostic approach, and C, fire, where we try to purify and transform the unstructured data into structured one, and verb, wind, where we try to control the reactive oxygen species generated by our own cell's battery mitochondria, and Yang, where we try to make use of the meso or the nano size space for detecting the modification that happens at the nucleic acid level. Let's dive deep into our elements and hear it from them.
Hi, I am Fujita, B3 student. I am working on the data processing tasks on the nanoprocessing project. Genome sequencing is a huge amount of data, which is like a chunk of iron ore. I write computer programming script as a blast furnace, where I burn them into human visible legitimate information. My future goal is to design algorithms to extract meaningful patterns from big clinical data set, guiding clinical decisions. Other than playing with computers, I also play the bass guitars.
Hi, I am Yuki Kobayashi, a B4 student. I'm currently working on a detection method for nucleic acids. qPCR is the gold standard for nucleic acid detection, but in recent years, innovative detection methods have been developed, such as RAN and RPA with new tools such as CRISPR-Cas12a and Satin-A. However, in spite of so many detection methods, there is currently no rapid and sensitive point-of-care detection method that does not require any special equipment. So I focus on lateral flow and hybridization chain reaction called HCA. I'm currently working on developing this technique for clinical application. Thank you.
Hi, I'm Dr. Vajanti Sandavar, working as a research scientist. My expertise includes organic synthesis and biocatalysis. Now I am developing the chemical compounds that can regulate reactive oxygen species generated by mitochondria in live cells.
Hi, I'm Yuki Shimiz, B4 student. I'm involved in the biological aspects and data processing. In previous research, it revealed that RNA in cells are folded intricately and it plays important roles on translation. I tried to detect RNA structure called as duplicate. Thank you.
I'm Soundar. I work on naturally occurring polymers called RNA. These RNAs are made up of 4 building blocks denoted by A, U, G and C. Inside cell, these building blocks undergoes various chemical modifications which are crucial for normal development. At times, aberrant changes to these chemical modifications leads to disease like cancer. We in Namasivayam Group are interested in identifying these chemical modifications using chemical probes and nanopore sequencing. Nanopore is a pocket-size sequencing technology, a disruptive innovation which is revolutionizing the field of RNA genomics. We are interested in designing nanopore-based methods for detecting chemical modifications as disease biomarkers. Apart from probing RNA modification, I am interested in cycling and cooking for my friends.
Hi, I'm Shubham Mishra and I work on epitranscriptomics. And what we basically do is my colleague helps generate long-read sequencing data from RNA using Oxford nanopore technology, and this data is typically in the form of electric current signals. What's important to remember is that this signal contains a lot of information about the chemical nature of the nucleic acids that it was derived from. And so it then becomes our job to analyze this data using pattern recognition and learning techniques implemented in common scripting languages such as Python and R, and also utilizing tools and scripts written by other bioinformaticians to extract relationships and patterns. By doing this, we then identify where in the sequence we have these modifications, and that's what we're trying to find out. Thank you.
Hello, I'm Abe Katsuko, D1 student, and I'm now working on the crystallography and the construction of the nanoprobe to detect the messenger RNA. And about crystallography, I'm conducting X-ray crystal structure analysis of DNA binding small molecules. And about developing probe, I'm trying to make the on-demand probe using silicon nanoparticles to detect the targeted messenger RNA by each patient cells without lysine. And it's for the first time for me to use silicon nanoparticle. So if you have interest and some advices, please feel free to talk and discuss with me. Thank you.
Hi, I'm Sangha. In Japanese it means mountain and river. I did my PhD from the Graduate School of Engineering, Kyoto University, and now a postdoc at the Namasivayam Group. I love working at the interfaces, interdisciplinary research, which also happens to be the founding convictions of the ICMS. I also have an industrial R&D background where I specialized in the process development for the purification of therapeutic proteins. I'm currently investigating the gene expression analysis of DNA binding molecules at single-cell level. Single-cell analysis allows us to study cell-to-cell variation within a cell population. This information is important for cancer research in detecting rare tumor cells, pre-implantation, and genetic diagnosis. I collaborate with my lab members in developing antibody-based detection methods to map epitranscriptomic modifications, along with providing analytical solutions. I also team up with different groups at the ICMS in developing microfluidic approaches to explore electrokinetic behavior and investigate the mechanics of DNA reprogramming. Apart from research, I'm a meditation enthusiast and I'm quite conscious about taking long exhales.
Hi, I'm Dr. Rihanna Ziadlu, a GSPS postdoctoral fellow who will be visiting Dr. Namasivayam Group soon from University of Zurich, Switzerland. I'm going to research on programmable control over inflammation by using smart genetic switches. My hobbies are skiing, hiking, swimming, and cooking.
Hi, I'm Dr. Valentina Vasoli, a GSPS PD who will be visiting the Namasivayam Group soon from our research institute, Davos, in Switzerland, on the Alps. I'm going to research on a neurogenetic marker for the identification of new topics for osteoarthritis. My hobbies are reading, visiting museum, playing guitar, and violin. I'm looking forward to this nice experience, new culture in a new place. I'm looking forward for it. See you soon.